Welcome back to the Late Late Show with Bella Watson. I'm your host this evening, and today our guest is world-renowned virologist Emily Ding. Thank you so much for having me on your show, Bella. So, you're a virologist. What exactly does that mean? Well, so basically most virologists study viruses that affect humans, animals, insects, or fungi, or just in general any organism in any type of environment. So since I am a medical virologist, I focus more on carrying out clinical research on relating to... Okay, so what types of education and training did you have to go through to become a virologist? Well, I attended Baylor College of Medicine in Houston, and that's basically where I got my PhD in virology. I also did some biochemical laboratory training on the side at Houston Metropolitan Research Center. Now, I don't mean to be too invasive, but how much is your salary? Since I work in Texas, I make around $53,000 a year. Alongside that, I do garner additional salaries from facilitating and monitoring university labs. Wow, okay, so I know you've been doing some research for the past few months. Would you mind telling me a little bit about what it is exactly you're doing? Well, as we most commonly know, mono or infectious mononucleosis is a prevalent viral disease that infects a wide range of people across the globe. Specifically, the EBV virus is a human herpes virus that is basically capable of infecting 90% of the population. Although primary EBV infections produce no symptoms of infection in early adulthood, but the latency of this disease is often associated with lymphoproliferative diseases. And if you're not really familiar with that and you don't really know what that is, these diseases rapidly basically divide, and these lymphocytes, which are the cells within the lymphatic system, multiply without control in excessive quantities. Usually our body's lymphocytes eliminate foreign antigens by releasing antibodies, and however, as the lymphocytes rapidly, a case of lymphatic cancer is developed. Now, how exactly does the EBV virus lead to cancer? Alright, so a great example of understanding how EBV can lead to cancer is through Hodgkin's disease. Hodgkin's disease actually has a threefold increased risk, and in order to test the relation of EBV and Hodgkin's disease, we can take a biopsy sample from the Hodgkin's disease tumor, and within those cells that there are lymphocytes that are actually abnormally large under the light microscope, and those are also known as Hodgkin and Reed-Sternberg cells, or just HRS for short. The latent EBV infection results in the expression of viral oncogenes, which contribute to generate the particular phenotype of the HRS cells. EBV also basically just induces epigenetic changes within the host genome. The presence of EBV alters the composition and the activity of immune cells, and by doing that, those immune cells will surround the HRS cell. In fact, the biopsy of the HD specimens identified a particular gene called EBNA1. So basically, as an mRNA, it will initiate transcription of the viral genome from the EBV virus. So by doing that, we found that the EBV viral DNA undergoes transcription, and then it will replicate and conduct maintenance of the viral episomes, or just plasmids for short, within the infected cell. EBNA1 gene must be constantly expressed, and actually the RNA data shows that the EBNA1 is expressed constantly at a protein level within Hodgkin's disease, showing its high level in expression, of course. As the viral genome is constantly replicated by EBNA1, the same genome can be found in the Hodgkin lymphoma tumor cell. So that kind of connects its cancer and the EBV virus. Wow, that's so cool. Isn't this research developing into a product? How is that going along, and how do you hope that this product will prevent the development of cancer? Upon finding antibodies to EBV by using infected patients' blood serum, we isolated 5 antibodies, but only 1 out of the 5 antibodies, known as AMM01, accomplished to successfully completely block the infection in the infected cells. In fact, not only did AMM01 block the infection, there was actually like 2 different types of cells being tested, and they were both infected with EBV, B cells and epithelial cells respectively. And with AMM01, they were both blocked. So it is capable of doing this because AMM01 binds to a viral protein that causes the neutralization of both cell types. So by successfully blocking the infection in both cells, we are confident that incorporating this AMM01 antibody into creating a EBV vaccine will ultimately end the development and relation of EBV virus to cancer. I want to thank you guys for watching our show, and my voice is dying, and I don't get paid enough to do this. Good night! Bye!